I will give the next presentation myself, if we can get it up. So I will talk about development of nanoparticles for clinical use and stress the importance of degradation and excretion. And what I'm going to tell you is very influenced with my background, since I have been working more than 20 years with heading metabolism, biodistribution, and excretion of all type of contrast agents in Nycomed, which was bought by Amersham, which was bought by General Electric, and been involved in bringing 5 products to the market and another 5 into clinical trials. And I left the industry some few years ago, and now working in the group of the previous speaker. So I think in order to improve the field and to bring more particle more rapid into clinical use, we have a work to do to improve the quality of our biological studies. It's also necessary to involve much more cross-functional collaboration. But one important issue I will shortly stress later in the presentation is that there are so large differences for different nanoparticles regarding how much studies you need to perform, the risk of failure, and the cost of development for different types of nanoparticles. I think that is a very important issue. I will cover it shortly, but we have discussed it in this article in Nanomedicine a couple of years ago. So, after intravenous injection of nanoparticles into blood, these particles have a very restricted ability to exit blood. They can pass through fenestra, small holes in liver, spleen, bone marrow, kidney, and there may be leakier endothelium and inflammation areas and in solid tumors. And very small NPs can be excreted through kidneys. And regarding how small or large particles then can be excreted in kidneys, I refer to a very good study published 10 years ago. And this study is often referred to as nanoparticles of 5 nanometers. They are rapidly excreted in urine. I don't think that's a good conclusion. It's much better to say you have a rapid excretion of approximately half of the particles when there are approximately 5 nanometers. And if they are larger than 10 nanometers, there are practically no urine excretion. And that's why it's so important that these particles are biodegradable. They need to be degraded in order to be excreted. And that's very important both for safety and regulatory issues. This may, of course, not be a problem for many products based on liposomes or albumin-based product, endogenous-based substances, or for iron oxide contrast agents that have been used for many, many years. They are known to be biodegradable. But in fact, there are very few data available for degradation of other nanoparticles. Very few. So, regarding targeting of nanoparticles, we have the passive targeting through fenestra and uptake in res. We have heard a lot about EPR during this meeting. And regarding active targeting to receptors or cells, we should remember that they are not target seeking. I think that selective retention is, in fact, a much better word than targeting. But how much of the injected dose is ending up in the target? We have also had some discussions about that in this meeting. You often see data like this, that you have a certain enhancement after doing something, but absolute numbers are very seldom given. And the only data I am aware of regarding such studies in humans is with radio-labeled liposomes that were made to be exactly similar to Calyx or Doxil, and where it was reported then in 2001 that you have 0.5 to 3.5% of the injected dose in solid tumors. And people are then speaking about, can we improve by using targeting principles? Well, with my background from medical imaging, I think it's worth to mention that we know 20, 30 years ago that by radioactively labeled antibodies, much less than 1% of the injected dose homed into tumors. Much less. So we cannot expect a huge increase in the amount of substance homing into a tumor by using targeting molecules. But even a small increase in binding may still be important for binding and uptake and the final efficacy. When speaking about targeting, I would also like to stress that you can often see in journals that people have done something that they call targeting, see an increase, and conclude that we have obtained a huge increase by using this targeting. But you, in most cases, see that they lack the necessary controls. It may just be that they have changed the surface properties, charge, and so on, and that's the reason. To measure distribution, metabolism, and excretion is not, in fact, a very straightforward issue. And you need to think about that you are not only measuring the drug. You need to know if the drug is encapsulated in the nanoparticles in an inactive form or if it's released. And this is not a simple task. So if you are interested in this, I think you should look into this publication where the last speaker in this session, Scott McNeil, is one of the authors, where they used the so-called stable isotope method to analyze both what were inside the particles and released. People are also using a lot of different type of labeling, maybe fluorescent or radioactive labeling. I would just comment that here with an example of radioactive labeling. It's important that you know that what you measure is really bound to what you would like to measure, so you are not lost the tracer. And you have the possibility to measure biodistribution in small animals with quantitative whole body autoradiography. It's an excellent principle that you can use tritium label of one compound and 14C for another, and if you combust it in a sample oxidizer, all radioactivity is separated into two different vials in water and CO2, so you have no tissue quenching. But the issue is that all these methods, they are not very commonly available, and they are all very laborsome to perform. It's a lot of work. Something about documentation needed for market approval of drugs. Times don't allow me to go much into detail here, but of course you need to document all constituents in your product. And, very important, the reproducibility of the production process. And of course, if you make products more complicated, you increase the cost for this analysis. You increase the risk that you will fail. Regarding safety and toxicity studies, I will just comment upon that if your particles are slowly degraded or slowly excreted, you need to perform such studies for a much longer time than for something that is rapidly degraded and excreted. And what is much longer time? Well, it's not easy to answer. There are guidelines to describe this, but they are nothing more than guidelines. And I think that in each case, you just need to discuss with the regulatory authorities. I'm just also commenting about this on my field, the distribution, metabolism, excretion. I'm really wondering how much data will regulatory authorities ask for when it comes to nanoparticles, because it's not much that has been published and it's clear about that. Regarding cost of development, it's cost a lot for both contrast agents and therapeutics. I haven't seen any data when it comes to nanoparticles. But what is clear, that more constituents means more analysis and more studies and higher cost. And it will certainly be a higher cost of trying to do something with a non-endogenous substance than with endogenous. And it will be a higher cost for a non-biodegradable than a biodegradable, no doubt. If you think you have a super nanoparticle and you would like a company to go to develop that further towards the market, you can expect that the pharmaceutical company will have a lot of questions regarding risk-benefit evaluation. What about the market potential? Better effect or less adverse reactions than product on the market? Expected cost and time for development? Risk of failure? Risk of not satisfying formal regulatory demands, for example, lack of reproducibility of batches? Expected manufacturing cost versus market price? And very important, the company is not just interested in the current situation. They need to think about what will it be when they have developed the product and is ready to go marketing. What's the risk of companies coming to the market with an equal, cheaper, or better product? It's, of course, extremely difficult to foresee, but they need to think about it. Some words about Theranostics. Many researchers focus on the possibility of making Theranostics, agent that can be used both for imaging and therapy. There's absolutely no doubt that that is very useful for in vitro studies and for animal studies. I'm not sure when it comes to clinical use, because it will be much more expensive and complicated to manufacture. Drugs must be approved for each separate use. Optimal properties are most likely not the same for therapeutics and imaging agents. For an imaging agent, it is very important that the agent is rapidly excreted, because it's really the signal in the diseased area compared with the surroundings that are the important. And for therapeutics, you should expect that it will be better if the particles circulated for a longer time. So I think that it's better to focus upon agents for one specific use. And if there is one thing I should try to summarize after 25 years in the pharmaceutical industry, it is make products as simple as possible. Just at the end, stress the need for cross-functional collaboration. And as the previous speaker said, we have a poster describing this, and I'm just standing up also with the same image of the group. And thank you for your attention, and we'll be happy to take any questions.
One question, could you explain why their contrast imaging agents development costs less than the therapeutics? What is the reason? It's an impressive difference. Huh?
It's many differences there. And one thing is, of course, that for many contrast agents, it's expected just to be injected once. Whereas for therapy, you also need to show a much more complicated effect, and by having an effect, it's also a danger that you have an adverse effect. And something that probably contribute to this is that, for example, speaking about the X-ray contrast agent, they are extremely inert substances, and although the dose injected is very, very huge, almost everything is out in urine in 24 hours. So it make it much more simple to follow up.
Thank you.
So what do you think is the best preclinical model to study ADME of nanoparticle? We should use different species or focus on the rodent model that is better characterized?
Yeah, we normally prefer to start with small animals like mice. From a background in pharmaceutical industry, we normally used rat and dog, if not any very specific case that we need to go to primates, but very seldom. But you need to have two different species and not two rodents. Of course, then, when using small animals, you have less to discriminate in an image, but still I think that the fluorescence imaging, MR imaging of small animals, PET-CT of small animals, they also all offer good possibilities. But regarding the measurements or biodistribution, metabolism, and so on, I think that you need to select your strategy based on what type of product you have. And the best thing would be to combine different approaches. And very important, once again, what I stress, that if you use a tracer, that you try to document that the tracer is still bound with the substance at the time of quantification. I think I've seen several examples in literature where that is not the case. Very short question.
Short question. Well, actually two, but one.
I think we need to stay to one.
Okay, just one. No, regarding what you mentioned about targeting, that is of the amount of the drug that actually, or the particles that actually reach the tumor site. We have several targeted molecules already approved in the market. One example is that TDM1 for breast cancer. It's the antibody attached to the chemotherapeutic. And when we use a nanoparticle, we usually have several targeting molecules on the surface of the nanoparticle, plus a large loading of the drug inside of the nanoparticle. So the results that we will see are expected to be different, but in cell culture, there's nothing interfering with the ability of the targeting molecule to reach the target. I believe some of the issues here are the blood flow and all the mechanics, like one of the talks that we saw in the previous session. If you could comment on that a little bit.
I just very short that I think it's much easier to get targeting in a cell culture than in vivo. and you often see very nice in vitro data with cell cultures, but when people start to inject in animals, it's much more difficult. I think we have to go on.